Well, hello, everyone. Good morning. Happy hump day. So today we're going to dive right into genetics. And again, we're going to focus on the aspects of this topic that are things you may not have covered before. With that said, we're going to do a little review and kind of make sure we're all on the same page. So in the past, what you will have learned is eukaryotic genetics. I'm probably focused on gene expression, right? How DNA is copied, replication, and how it's expressed, how it is transcribed and translated into proteins. What we'll focus on is the differences between prokaryotic and eukaryotic genetics that stem really from the cell structure differences. And we'll also talk about some specific examples of gene expression regulation that are found in bacteria. So as you know, I always like to sort of start off with why we should care, okay? And I will just sort of cut to the chase here and say that why we should care about genetics, which is a topic that not everybody is in as much adoration and love with as I am, is because it underlies all the other things, such as metabolism, right? Because what you're able to do metabolically is determined by enzymes, and enzymes are genetically encoded. It underlies cell structure because the cell structures and functions, the way the cell is created, what it's made of, how it works, what it does, what the different parts do, is all relying on proteins, which are genetically encoded, right? Even the parts of the cell that are not protein-based, like lipids and carbohydrates, are created or organized and assembled by enzymes and proteins, or they're made of proteins. And then things like industrial applications, things like manufacturing, or things like how does this microbe contribute to the ecosystem? That, of course, is all about its metabolism, which is all about genetics. So let's get a little bit specific and kind of, I hope that I'll surface and help you appreciate that you've actually learned some things in this class. So what are some features of microbes that are determined by proteins? You can think about this in either of two ways. You can think about like physical features that are protein-based, okay? So what things in chapter 4 did you learn that cells have, particularly bacteria, which was our major focus, but it could be eukaryotes too, that are made of protein, and specifically what are those structures, or what are things that microbes can do that rely on proteins? So just put those answers into the chat real quick and I'll call them out. Yeah, motility, right? Motility is based on the ability to have some kind of protein-based flagella or fimbriae or pili in bacteria or cilia in eukaryotic microbes. Synthesis, yeah, what can they make? That's based on enzymes. Enzymes themselves, what can enter and what can't enter the cell, right? That's based on transporters that are in the membrane. The process of respiration, can they do respiration or can they not do respiration? What form of respiration can they do? Different cell structures, their ability to attach, right? Whether they have the protein pillin or don't have the protein pillin. Can they attach to things or not as bacteria? Pathogenesis, okay, so what specific protein structures do they have that make them more or less pathogenic, which we'll go into more detail in the pathogenesis section of this course. So many of you have mentioned some of these. Here are some other ones in this list as well, right? So what type of cell wall do they have? That's determined by enzymes, okay? And this is related to their antibiotic sensitivity in some cases. Do they have a capsule? Capsules relate to their virulence. Are they as dangerous or less dangerous to us? And the capsules, again, while they're made of carbs, are created by enzymes. If you don't have the right enzyme to create the capsule, you don't have a capsule. Flagella, right? And flagella relates to, are they motile? Can we help in some ways to identify who they might be? Flagella are made of protein. In bacteria, they're made of flagellin, and in eukaryotic microbes, they're made of microtubules and motor proteins. Can they use specific nutrients? What kind of exoenzymes can they secrete? Are they able to do fermentation? For example, we rely on this for a lot of our food production that relies on microbes. And can they survive in oxygenated environments? Okay, so these are just a small list. I mean, we could go on and on. So genetics helps us do two other things related to what we've talked about, but kind of getting into bigger picture things here. Looking at genes, reading the specific sequence of the genes, that is a relatively new thing, right? It's as revolutionary to the field of microbiology as the electron microscope was to the field of cell biology. But now we can do that. It's like that genome is a fingerprint. We can establish exactly who we're dealing with. So we can identify organisms very specifically. And also we can sample environments in a genetic way without sampling it by culturing things necessarily, which can be problematic, right? You don't know exactly what they need to eat, so you can't grow everybody. You can sample pretty much everybody genetically. You don't have to grow them up and know what to feed them to sample their genes, because we can take very small amounts of DNA and make lots of copies of it and read that sequence. So it gives us a window into the diversity of microbes, which turns out to be much greater than was originally thought. Genetics is important because microbes change, right? They go through changes, and this has lots of implications. But in terms of sort of like maybe the pathway that you're on, genetic change underlies the development and rise and spread of antibiotic resistance, and related to that, the arising of new pathogens, in particular viruses, but other things too. And then we can actually alter the genes of bacteria to make them useful for us. So we're going to start with a really quick, I think, review and somewhat superficial review of the type of genetics that you likely studied before, which is how do you copy DNA, the process of DNA replication? How do you express it, the process of transcription and translation to give rise to proteins? And then how does it change, which will include some new things. And we will focus on prokaryotic processes for the most part, but we will also compare eukaryotic and prokaryotic cells. And we're going to start with the copy, okay? So this is a picture of bacteria going through cell division, which requires a DNA replication event. In the past, you will have studied cell division as mitosis, most likely, but it still had to go through a process of DNA replication before the cell could divide. And that turns out to use the exact same enzymes in all cells. Like all cells pretty much across the board use the same process to copy their DNA, even if the cell division part afterwards is different. Now, here's a list of the major enzymes involved in DNA replication, not in the right order. Your job is to look at this list and put numbers in order. Okay, so write into the chat if you think this is already the right order, you just write 1, 2, 3, 4, 5. If you think it's backwards, 5, 4, 3, 2, 1. But probably that's not how I did it. So put the numbers into the chat in terms of the order in which these enzymes are required to get DNA replication started and then completed. And I'll give you a second to do that. Great. So lots of good answers rolling in. I love that everyone has agreed on a lot of the early starting enzymes. A little bit of questions around sort of the maybe finer details, but for the most part, everyone's on the same page here. So, we start with DNA helicase, RNA primase does its job next, DNA polymerase then comes to work. DNA polymerase 1 is follow-up to this, which may be a nuance you were not aware of or don't remember, and then DNA ligase. And I want to be real clear that there's not going to be like a test question where it's like order these enzymes. I'm just trying to get us to the place where we can surface and remember the things we know so that we can lean on it for the rest of the curriculum. So I'm going to cold call you to tell me what the functions of these are, okay? So I'm going to give you a minute to think about it. And I want to remember, and I'm really sincere when I say this, if you don't remember or you don't feel comfortable venturing a guess, passing is fine. But with that said, don't be afraid to guess. We're just trying to get, there. Okay, so I'm going to start with Sydney. What, if you know, is the function of DNA helicase, or what does it do in this process of DNA replication?
In my notes, I wrote down denature.
Yes, it denatures the two DNA strands, right? It breaks those hydrogen bonds so it can pull them apart so they can be each copied. Let's go to Alyssa next. What is the job or the function of RNA primase?
I think I'm going to pass.
Not a problem. Thank you for doing that, Alyssa. You made everyone else take a big sigh of relief this morning. They're like, oh my gosh, yay, I don't have to be so worried about passing. Spencer, would you like to take up the banner and tell us what RNA primase does, if you know, or you're welcome to pass.
I will pass.
Let's see. How about Andy? And if you're able to unmute, go ahead.
Hello?
Yep.
Sorry, I would like to pass. Sorry.
No worries. This is a harder one. So what I'm going to do now that I put everyone on the hot seat and everyone's been like, oh my gosh, I had to pass, is raise your hand if you know the answer to this one. Let's see if anyone does know it. Mackenzie, go ahead.
It primes for DNA polymerase 3.
That's right. And what that means is it is an RNA polymerase itself. It actually makes little pieces of RNA. So it will start the chain by making a series of several connected RNA nucleotides that match the DNA in the place where replication is going to start. And it kind of sits it there like a denture, right? So that DNA polymerase can do its job, okay? And before we say what that is, let's put someone on the hot seat. Aubrey, any thoughts as to what DNA polymerase does once RNA primase has primed the chain?
I'm going to pass on this one.
Not a problem, thank you. Riley, you're up next. What would you say if you know, or pass is fine for this DNA polymerase?
Doesn't it just duplicate the DNA?
That's right. So DNA polymerases in general are not able to start chains. They can't prime their own chain. That's why RNA primase has to do that. But once there is a little bitty chain started, they can just hook extra nucleotides to it. And that's what DNA polymerase does. It follows the template of the DNA strand it's trying to copy, just keeps adding nucleotides. So it extends the chain. Now, this is a subtlety. So rather than put everyone on the hot seat for this, does anyone know, raise your hand if you know what DNA polymerase 1 does? After we've gotten the DNAs open, we got the chain started, we got the chain extended. Sam, what does DNA polymerase 1 do next?
I think it does, it just looks at the chain and it makes corrections and fills in gaps and fixes things and things of that nature.
That's totally on the right track. It's a little bit more specific, and this is not something I'm going to test you on, nor do I'm expecting that everyone came in knowing this, but it comes back and it looks at this little piece of primer. So the beginning of the chain is actually made of RNA, because that's what RNA primase makes. And that can't stay. It's not like you get this, like, you don't have DNA that's got little sections of RNA in it. So DNA polymerase comes and removes those little pieces of RNA and replaces them with DNA. So it is a fixing of, but it's a specific fixing. In this context, it has other roles in other contexts. Now, this does still leave an issue unsolved that DNA ligase solves. And let's go to Misty. Do you know what DNA ligase does? It's also
Can I pass?
You can, absolutely. Quinn, do you want to take that up?
But it's kind of a guess, but does DNA ligase like connect the fragments?
That's exactly right. So in the process of doing all this, there are little gaps in the sequence, not in gaps where like there's no nucleotides, but rather that there are stretches of nucleotides that are not connected to each other. And DNA ligase finds those and connects them. In particular, this happens on the lagging strand. You'll remember those Okazaki fragments, but it also happens on the leading strand. Okay, so again, DNA helicase opens the double-stranded DNA. RNA primase starts the chain because DNA polymerases can't do that. DNA polymerase extends the chain. DNA polymerase 1 replaces the RNA primer with DNA, and DNA ligase seals any gaps. Just to make sure that we also have a fairly accurate picture of the structure of and the biochemistry of DNA replication. Here's a DNA molecule. It's obviously got two strands. They are anti-parallel. You can see that there is a sidedness to them, right? That there's sort of, this has a a so-called 5' phosphate group attached to the 5th carbon of this sugar. And over here is a 3' hydroxyl group. And on the other side, the orientation of the matching strand is reversed. So what is the 5' end at the left side? I already said this, but if you didn't come in knowing this, you might not have heard it because it's very jargony, right? That's the 5' end. But is it the top with the phosphate group or is the bottom with the hydroxyl group? Yeah. The way that this labeling is assigned is this is a 5 carbon sugar. You remember this is deoxyribose, a 5 carbon sugar. And the first carbon, or the 1' carbon, is the one that's attached to the nucleotide. And then they go around the circle in a clockwise direction to assign the numbers of each of the other carbons. So that's the first carbon, second, third, which is where the hydroxyl group is. Fourth, fifth carbon's right here. It's attached to the phosphate group. Okay, so if you were to be adding nucleotides to the end of the left side, okay, so if you opened this up and you started to copy it. So let's say this is your template and this is the copy, okay. Would the next nucleotide be added at the bottom or at the top? And if you don't know, putting a question mark is perfectly fine. Great. So it's this 3' hydroxyl group, which is where there's a useful chemical side group that the next nucleotide can hang itself onto. You'll remember if you've done the biochemistry review module that it is a dehydration synthesis reaction, right? So the next nucleotide kind of looks like this, except it's got a hydroxyl group of its own. And the phosphates have some hydrogens. And so one of the hydrogens from the phosphate group pairs up with the hydroxyl group. They go off as a molecule of water. And then those unsatisfied bonds grab onto each other. Here's a picture, a little cartoon of DNA replication. Which letter represents the leading strand? And just for reference, sometimes people use leading strand to refer to the parental DNA, and other people use it to refer to the daughter DNA. I use it to refer to the daughter DNA, the newly synthesized DNA. So there's two options, really. Which letter represents the leading strand on the side of the newly created side? Yeah, it's obviously B. And what makes it the leading strand is that you can see the orientation of it, because you may remember that these enzymes have a particular direction they can go, right? Remember, we just talked about how you have to add your next nucleotide to the hydroxyl group. And so the 5' end of this newly created daughter strand is over here. This is where the phosphate is of the first nucleotide. And over here, this last nucleotide that was just added has a free 3' hydroxyl group. That's where the next nucleotide is going to go. And so once you start this chain over here, it just can keep going forever. But on the other side, this strand is being copied at the same time alongside this, but because it also has to go in the 5' to 3' direction, and it's the opposite orientation, then it has to be copied in this direction while everybody is also going this way. So a while ago, this was laid down, this first fragment was laid down, and then this was created next in this direction, and this was created next, and this is a new one just getting started. This lagging strand is lagging because it's a little bit behind. Okay, which letter points to helicase? Yeah, hopefully that's an easy one. Right here it is, unwinding the strands, and it's moving in this direction to the left. That would make the orientation, or not the origin of replication somewhere over here, like on the right hand side. Which letter indicates the enzyme that makes primers to start the chain? Yeah, it is C. All right, it's laying down this little pink part, right? The primer. And so what's the name of this enzyme? Yeah, RNA primase. That's right. This is RNA primase laying down a little piece of primer. And what's the primer made of? At the end of the day, when a replication is completed, everything is made of DNA. What's this primer made of? Exactly. It's made of RNA, right? It's made of RNA because this is an RNA polymerase. It's making a little piece of RNA. Later is going to be this little pink section is going to be replaced with DNA by DNA polymerase 1. Okay, which letter points to DNA ligase, the one that's sealing all those gaps? That's right. And you can see on this lagging strand, because we had to go this direction and then hop up here and start a new strand, that there is just a little bit of discontinuity between the strands, and that's the job of this guy, to seal those up. Okay, with that in mind, a couple questions. A bacterial cell that acquires a mutation that disables the RNA primase, so RNA primase enzyme is kaput, does not work. What would be the consequence? It would be unable to synthesize the leading strand, unable to synthesize the lagging strand, or unable to synthesize either strand. Correct. Both would be affected. The reason I ask that is these types of pictures, these static pictures, they sometimes obscure the totality of what's happening. Okay, I think it's pretty easy to understand once you see this picture, and if you can remember and kind of diagram this picture, it's easy to understand that RNA primase is required for the lagging strand. Okay, because we can see it's being needed to prime this one and prime this one and prime this one and prime this one. And there's no, in this picture, it doesn't seem like it was important. But where did you need this RNA primase on the leading strand? Yeah, at the beginning of this guy right here, right? Somewhere over here, before this picture starts, there was the origin of replication where this whole shebang got started, and we did need to prime that to begin copying. Yeah, exactly. And so here's a little picture of that, kind of looking more fully at the beginning of this whole process. There's the origin. That's where all the machines assemble. Here's helicase, but it's running ahead of the whole deal. And the very first time you made anything for the leading strand, you had to lay down a primer. RNA primase did that. Then there was a hydroxyl group. DNA polymerase was like, sweet, now I can get going. And it started adding DNA. So this is all one continuous piece, but it started with RNA. On this other side, of course, we've got primer DNA, and as this moves to the left, we're going to primer DNA, and all these little gaps will need to be filled in with ligase. Now, I want to turn our attention to, so that's basic replication. That's true in all cells, okay? But in bacteria, one of the things that's different than eukaryotic cells is that there's a simpler DNA, and it's a circle, right? It's a circle and there's one. And of course, we're also replicating in a cell that has no nuclear envelope, no microtubules, so no spindle, no centrioles, no mitosis. So all that you need to do is basically make your copy of DNA and then literally hang each copy on a little protein hook on each side of the cell and then just build a cell wall in between them. Now, in addition to these differences, that there's this smaller single chromosome and it's circular and there's no nuclear envelope and no mitotic spindle. Something that is true is that where things begin is the same. What do we call the series of letters in DNA that tells all those helicase, primase, polymerases where replication should begin? Great. Now, I'm really pleased with your answers, and it's not because they're correct. They're not. But I'm pleased with your answers because this is a common misconception and confusion that people have. And that's across the board. So there's nothing to feel bad about. But it's really important to kind of face that head on and look at it and go, can I remember that this isn't true? So everybody says this. Everyone's like, well, the start codon, of course, right? The start codon, of course. But the start codon is actually something that's found in messenger RNA that talks to ribosomes. And sure, the letters cryptically are present in the DNA, but it's in the DNA, it's not telling anybody anything. It's only when it's made into RNA that it becomes informative. So actually the start codon is not where replication starts. It's not even where transcription starts. It's the origin of replication. And the origin of replication has no relationship with any particular gene, or it's like where replication starts. And in the case of bacteria, there's one origin, okay? If it was the case that start codons needed to start replication, then that would be pretty confusing because there's start codons all over the place and lots of genes, right? So this is called the origin of replication. It's frequently abbreviated ORI for ORI, but this is what it stands for. And it's just a series of letters that is like, we think of letters as being sort of words, and I guess that's true, but they are also, they're like signs for the cellular enzymes, and the origin of replication is like a landing pad or like a flashing sign or an arrow that says to primase and helicase and polymerases, this is where to start replicating. So an important thing to understand, and this will take some of you a minute, okay, is that because there's no nucleus, and because the cell is so much simpler and doesn't require complicated mitotic spindle or mitosis or anything like that, we don't have to build a nuclear envelope or take it apart or separate multiple chromosomes from each other, then it is possible to do something called continuous replication. And that's shown in this picture. I'm going to do my best to explain it to you. But that's when there is a new round of replication that starts before the last one is finished. It starts about halfway. So here's our original molecule. You can see it's double-stranded. Here's the origin. We pull it apart. We start replicating. Now, both sides of this replication bubble have now become double-stranded, and the replication forks are moving in this direction. So replication is moving in this direction. This is about halfway done. Okay. So this part is unreplicated, and here's the two sides that are the same as each other. Eventually we'll finish and separate them. This picture represents this, okay? And so what you can see is this is the part that is unreplicated, and this is the two sides that are copies of each other. Now, continuous replication was discovered because of this picture. When people were looking at rapidly dividing bacterial cells, they would bust them open and throw them down on a slide and look at them with an electron microscope to find the DNA. And instead of finding a bunch of cells that had circular DNA that looks like this, they kept finding a lot of cells that had DNA that looks like this, where it seemed like replication was halfway finished, like way more than it would really make sense. And so what eventually this picture kind of helps people to understand is that in prokaryotes, when you are, let me see if I can, I think I have it in this. Oops. I think I have it in this thing. I'll come back to it in a second. This is the origin right here, and the origin is also right here. Once this is halfway finished and there's machines that are moving this way to continue replication, this origin here and this origin here are again used as a landing pad for another set of enzymes. Helicase, a new group, I guess, a new company of helicase, polymerase, and primase to start replication again. So in this picture, what's really happening is not just that replication is finishing in this direction, but that a new round of replication is starting here and here. So what that means is that instead of you copy and you get to this, right, and then the cell divides, you copy, you get about here, you start copying again while you're finishing. So actually, before you divide, you actually have two of these, and that's what gets inherited by the next generation. So this helped to solve another mystery, which is this, okay? If you do the logistics and time how long it takes to actually copy an E. coli chromosome, it takes 40 minutes. But the generation time, which is how quickly the cells can divide under ideal conditions, is half that. If you've done the prep, you understand why this is. So maybe you can raise your hand if that's the case and tell us in your own words, like, why that's possible. All right, no one is feeling particularly brave, which is understandable on a Wednesday. And this is a complex concept to explain, even if you feel you understand it. So I'm just going to do a little diagram again to kind of illustrate what I'm talking about. So here we've got the origins, right? And this is a chromosome that's halfway finished replicating. And at this halfway point, while the replication is still ongoing in these directions, a new set of machines is sitting down here and copying both sides. So what gets made by the end before the cell divides is actually two of these. So right before I move on, I just want to ask if that makes sense. Yes, no, or kind of. And be honest, I have a fix for if the answer is no, and that's fine. Great. So here's my fix for now. Just remember that because there's no nuclear envelope and no need to go through complicated separation of chromosomes, then in bacteria, replication is not just start, finish, and then you divide. Replication is start, get halfway done, start a new round of replication so that the chromosomes you're making are halfway completed before you hand them off to the next cell. When cells are born, they don't look like this. They look like this. And another way to sort of like just straight up memorize this is just to memorize that something called continuous replication is possible in bacteria because there's no nuclear envelope. And what it does in effect is it cuts the generation time in half. They can get their DNA replication done twice as fast. Now, maybe logistically this doesn't make sense to you because at some point in history, right, there had to be like an intact circular chromosome that hadn't yet been copied. And to copy that completely will take 40 minutes. So that very first generation is going to take 40 minutes. But after that, if what you're handing down, if what is inherited by a cell is this, okay, if what it inherits is a chromosome that's already halfway finished, then it's only going to take 20 minutes to finish this part. And while that's happening, okay, the 20 minutes it takes to finish this part, if this is what you inherit, the 20 minutes it takes to finish this part is going to give you 20 minutes to do this part so that you can give this whole thing to your children. Does that help at all? Yes, no, or kinda. Great. So the thing to memorize if you're struggling with this is what Alyssa said in the chat is a nice summary. A cell can replicate before, a cell can start a new round of replication before the first one is complete. And it's going to hand off not a simple intact chromosome to its daughter cells, but a chromosome that is a 1.5, basically. It's 1.5 times. Okay, so we can revisit that if necessary. But really the take home that's important to know for like a test is that a prokaryote and eukaryotic cell, given the same amount of DNA, which would be unusual, but let's just postulate that, right? Giving the same amount of DNA, the bacterial cell is going to be able to divide basically twice as fast. So its generation time is going to be shorter if everything else is equal. Great. Awesome. Okay, so what are we going to talk about now? Now we're going to talk about gene expression. We talked about how to copy the DNA so you can pass it down to your kids. Now, while you're alive, what you're using the DNA for most of the time is to make proteins. And in fact, actually, gene expression is bigger than that. It's not just making mRNAs and proteins, because some of the RNAs that are made by transcription are not protein coding. They might be tRNAs or ribosomal RNAs or other things. So there's lots of genes in here. We got to express them. And we're going to talk about the basic mechanics of this. And then we're going to spend some time talking about in bacteria, the regulation of that gene expression, like when are these genes expressed and why, and how do we control that? Or how does the cell control that? How does the bacterial cell? We can control a little bit once we understand how it works. So this again will, I hope, speak back to things that you understand and have learned before, but let's just review them. To get from DNA to protein, you know, the sort of like very iconic version of genetics, sort of the simplest level of genetics, to get from DNA to protein, we have to go through 2 steps. We've got to go take the DNA and copy it to a messenger RNA, and then we've got to take that mRNA and get it somehow made into protein. So the process of going from DNA to the mRNA, or really DNA to RNA, because it's not always messenger RNA that's created in this process, is called what? What is that process called? That's right. That is transcription. And that's the first thing we're going to talk about. Now, if it is a protein coding RNA, if it's a messenger RNA, it then gets somehow used to make protein. And the process of using that, reading that RNA in order to make protein is called, yes, that is translation. Now, just a moment here, because this genetic section is particularly bad for this reason. There's a lot of words that start with these 5 letters: transcription, translation, transduction, transformation. And so it's easy to mix them up if you're in stress. And I just want to give you a way to think about it if you're having that moment of, I'm blanking on a test. What is she talking about? So transcribe comes from the word scribe. Scribes were people who, before the advent of photocopiers, they would literally copy books by hand by writing. They would read the book and they would copy it. Okay. However, in the process of transcribing, you can imagine they might be copying from an old book, and they might be, maybe the language has changed a little bit. So now instead of S's, they're using F's, or vice versa, or instead of V's, they're using W's. Okay. But it's the same overall alphabet. It's a letter for letter transcription, right? And that's how DNA to RNA works. This is the language of nucleic acids. You're going from, you know, AGCT to AGCU. Okay, you're just changing one letter, but it's essentially the same alphabet that you're using and the sort of same logic and everything. So you're just copying the book with a little bit of a spelling change. when you translate, you're translating between languages. You're translating from the language of nucleic acids to the language of amino acids. The alphabet is totally different. There's 20 letters in the proteins, and 3 letters of DNA are required to code for 1 letter of protein. So it's a different language. It's like going from like Russian to Korean, okay? Not the same alphabet. But nonetheless, there is a meaning. This means something, and you can make it mean something here. So hopefully that's helpful. Let's get clear on some transcription terms. So this DNA that we just copied, right? Somewhere in here, there's an origin of replication. There's just one. But there's a ton of genes in here. This bacterial chromosome maybe has 4,000 genes. So that means that there's a lot of different places where transcription needs to start depending on what we're making. So it's not the case that there's just one place where we start transcription and we just make a big mRNA of the whole thing. There's going to be, here's a gene, we're going to transcribe that. Over here's a gene, I'm going to transcribe that. So there have to be some other things, not origins of replication, but something that starts transcription. And of course, also, if we're going to do transcription, we need some kind of enzyme to do the job. So what is the enzyme that reads DNA and makes RNA? Great. So it is called RNA polymerase. And when we get to viruses, I'm gonna give you a better or more complete name for this enzyme. We also call it cellular RNA polymerase. We also call it DNA dependent RNA polymerase. And that is because once we get to viruses, the universe of polymerases opens up and we need to be more specific. But when someone just says RNA polymerase, what they mean is the enzyme that does this. Now, as it turns out, RNA primase is also an RNA polymerase, but we call it RNA primase because it's dedicated to replication. And we call this RNA polymerase because it was the first one. So it gets to keep the name. It's like, you know, you have a class and someone is, Eric is in it, and then a new Eric joins, but he has to be called Eric G or something. So this is RNA polymerase. When people just use this word, that's what they were talking about. But we will use a more specific name when we get to viruses for reasons. Now, as I said, there's going to be multiple places in the genome where transcription begins. Okay, this is the start of one of the genes, but there's going to be whatever this is, this sequence of letters that says, hey, this is where a gene starts. There's going to be a bunch of these. So what do we call this sequence? And just to cut to the, it's not the origin of replication because that is a sequence of letters that speaks to helicase and DNA polymerase. We need a series of letters that speaks to RNA polymerase and says, hey, this is where a gene starts. What is this series of letters called? Great. And some people have written the right answer. Some people have a different answer that's not correct. Some people have a question mark. These are all perfect. These are all perfect responses. You all have given me exactly what I need. Okay. So if you have no idea, well, that's understandable. It's just a jargony word. If you said promoter, good job. You're right. If you said start codon, excellent. Because again, this is something that's very confusing. It seems like it should be the start codon. It would kind of make sense, right? Because start codon is going to be helpful later on, but it's not the start codon. The start codon is actually usually right about here. So it's not even the very beginning. Like this part here is a landing pad. It's a series of letters in DNA that RNA polymerase lands upon. It doesn't even copy. It just lands here like a helicopter landing pad. And then it copies the stuff in front of it. And the start codon is actually going to be like buried in here somewhere. So this is called the promoter. And it's a series of letters that's the landing pad for RNA polymerase. Okay. And then eventually, because we're not trying to copy the whole thing, there's one gene. Okay. And there's going to be a series of letters. That's the end. It says, this is the start. This is the end. What is this called? Yeah, it's just like that iconic 80s movie that I grew up on. I'll be back. Terminator, that's right. You don't know what that is, then you are missing out and you are too young to have enjoyed the best part of life. So Terminator, and it's the transcriptional terminator. Now, there's other terms. We'll ask similar questions about translation in a moment, but for right now, let's have a quick look at an electron micrograph that's been pseudo-colored. And what we're looking at is transcription. It's a snapshot of transcription actually happening. Blue, it had pseudo-colored in blue is the DNA template. Pseudo-colored in pink are the RNA transcripts coming off of it and during transcription, okay? Where's the promoter? Is it on the left, lower left, or is it on the top right? Where's the promoter? Excellent. Yeah, it's on the lower left. And how did we know that? When you were able to say, boom, lower left. I'm just going to cold call a couple people. And if you want to say it was a guess, or I'm not sure how to explain how I know, that's fine. But I do want to hear what you were thinking. Libri, why did you say lower left?
Well, in the recorded lecture, I remember you talking about it with it.
Excellent. And what do you remember me saying, if you remember?
I don't remember exactly. I just remember you were showing us with the cursor, where.
Great. Excellent. Okay, perfect. Spencer, what is your, you started to give us an explanation. Spencer, tell us more.
Oh, it's just that things coming off of it are smaller on the left side. So that is probably the beginning.
Yeah, exactly. So the guys down here, so every one of these lines of pink, you have to know or imagine or understand that there is an RNA polymerase enzyme. You can't see it, it's really small, but it's right here. And it's what's actually making this chain. And it's moving in this direction to the right. And so down here, the transcripts that have been made by these guys are super small because they just got started, and the ones over here are pretty much already done. Okay, so there's the promoter, there's the terminator. The other thing that I want you to appreciate when you look at this picture is that it isn't like a zip line, okay? The tendency to think about this type of process is that the enzyme hops on and it makes its transcript, and then it hops off, and then it says, hey, I'm safe on the other end of the zip line, send the next person. And instead it's more like a free-for-all. Like we have enzyme grabs onto, lands at the landing pad, promoter, starts copying, zips down as fast as it can, but no sooner has it moved 1 inch than another RNA polymerase enzyme is right behind it doing the same thing. And then we've got thousands of guys. The reason that's important is to understand that when the cell is like, hey, I need to make this gene into protein or whatever, it's fast and it's huge response, right? It's not, you're not making one of something, you're making 100,000 of something, and you're doing it as fast as you can, especially if you're bacteria and you're trying to respond to a changing environment. And you're like, oh, shoot, I ran out of this food source. Now I need a different one. I need a different enzyme. I need it right now before everybody else eats this food and I die. Okay. Now, because there's so much jargon in the field of genetics, and it's not stuff that is necessarily part of your common language, or there are words that are, and then they just confuse everything. It's important to make sure we are straight in our head about the difference between the different processes. Okay, so let's see if we can kind of remember all the things under pressure. DNA replication starts where? Write that into the chat. Where does DNA replication start? Yes, good job. Correct. It starts at the origin of replication. And transcription, on the other hand, starts where? Where does transcription start? What's the landing pad or the enzyme? That's right. Good job, guys. See, you got it. It is the promoter. Whoopsies. Hopefully that's obvious. I'm just going to leave it up. In replication, helicase unwinds the 2 DNA strands. We remember that, right? There was that whole laundry list of enzymes. In replication, helicase unwinds the 2 strands. And in replication, who starts the RNA? Who makes RNA to start the chain? and be as specific as possible. So some of you have a more generic term for this, while not wrong, isn't as specific as you need it to be. That's right, RNA primase. So we're still in replication. We got helicase unwinding, RNA primase beginning. So is this true? True or false? Transcription also uses helicase and RNA primase. True or false? Great. It is false. DNA replication is a circus. You know, helicase, primase, polymerase, other polymerase, ligase. Transcription is a one-man band. Okay. RNA polymerase opens the chain, unwinds it, starts the chain, extends the chain, terminates the chain, goes on its merry way. Which of the following is the best description of the role of RNA polymerase? Right? Remember, when we say this, what we're talking about is that enzyme that facilitates the process, and it's not referring to, when we just use this term, it's usually not referring to any RNA polymerase, of which RNA primase would be one, but specifically the one involved in transcription. Excellent. Now, most of you picked C. That's not wrong, but it is not the best answer. The best answer is this one. Can you think about why that might be? It's certainly the case that RNA polymerase copies DNA into messenger RNA. But is that the only type of RNA that DNA can be used to make? What else is there in the universe besides mRNA that you still have to transcribe? tRNA, rRNA. Those are made from genes. They're just not protein coding genes. Okay. What about this? All the information in the DNA of a cell is organized into genes that code for proteins. So what this means, let me be clear. Every letter in DNA is coding for protein. This can be directly translated into protein. Yes or no? Excellent. So everyone is saying no, and it's a 50-50 on how confident you are. Some of you are like, well, I think it's no because of how you're talking about it. And some of you are like, I know it's no. If you know why it's no, raise your hand and help us understand. Sam, go ahead.
Some of the information isn't for coding genes. Some of the information is like the start and the terminator and stuff like that.
Yeah. So there's several types of letters in DNA that don't directly get translated into protein. And here are a few other examples. So the one that what Sam mentioned was like the regions that are informative to the RNA polymerase during transcription. The promoter region, I said, was a landing pad for RNA polymerase, but itself, that landing pad is not actually copied. It doesn't get made into protein. Furthermore, some genes are not protein coding at all, but they're still important to transcribe. So the genetic information that says how to make a tRNA or how to make a ribosomal RNA doesn't go to the ribosome and get changed into protein. It just already is what it is. The origin of replication is a landing pad for helicase. That's not protein coding. And of course, if we were talking about eukaryotic genes, there'd be like introns. You probably maybe sort of vaguely remember that. We don't have to worry about those too much in bacteria because they don't really have any, but introns is this, what people in the past have referred to as junk DNA that doesn't do anything, which now turns out to not really be that simple. Okay, so just some examples in this little diagram. If this is our double-stranded DNA, and let's say here's a protein coding gene. So this part, right, is going to be made into mRNA and then translated into protein, sure. But the promoter is not copied or translated. Here's the origin. It's not copied or translated. It's just informative for the machinery that needs to copy DNA. And here are some genes that have a promoter region and letters in DNA that get made into RNA by the RNA polymerase. But then what they do is they immediately fold into the shape there. Like this is the product. Okay, it's not an intermediate, it's the product. It gets folded into tRNA like a clover leaf to participate in translation, or it gets folded into some other shape that gets stuck into the ribosome. Okay, so I'm just going to go backwards for a second. Look at our time. And we have been talking for an hour. So I'm sort of feeling like maybe this would be, I can either do like 5 more slides and then take a break, or I could break now. What would you like? Do you want to just kind of blow through translation real quick? Or do you need a little second to kind of recover? Keep going, says Jolene, continue, says Autumn. Well, in my class, the first people to speak up get their way, I guess. So let's move on real quick. Okay, so here we are. We've made an mRNA in this example, and now it needs to be made into protein. And what is the enzyme / machine that's going to do this? What's going to read this messenger RNA and then spit out some protein? Yeah. Our good friend, the giant ribosome, indeed. Ribosome is a machine for doing translation. That's what it does. It needs a couple of things. Remember that ribosomes are made of protein and RNA. So they're made of protein and ribosomal RNA. The proteins are folded up into shapes like little squares and triangles and bolts and nuts. And the ribosomal RNA also is folded up into a particular shape, held together by hydrogen bonds, because you can do that with RNA. And they all fit together like 1,000 little pieces. That's the machine. And then the tRNAs, which are little cloverleaf structures, they come in, bring amino acids to the ribosome to match to the RNA code. Now, the start of translation is a 3-letter sequence called AUG. What is this called? This is where that answer y'all have been giving me all day is finally true. You're like, oh my God, when are we going to talk about start codons? Yes, that's the start codon. Notice that it is not the first 3 letters of this messenger RNA. Like theoretically, it could be, okay? And I'm not saying that there isn't any example of that, but it'd be pretty rare. So over here in the DNA, this AUG is an ATG, but it's not usually right here. And it's certainly not here. It's usually about here. And there's some kind of little like bit at the beginning that gets transcribed, but not subsequently translated. So there's going to be like, maybe this is UAGGC and then AUG and then blah, 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 and then whatever, okay? Then there's a 3-letter code at the end that indicates this is where we stop translation. What do we call this one? Yeah, this stop codon, and there are 3, right? Any 3. Okay, there's actually 3 stop codons. There's only 1 start codon. Last thing to remind ourselves about this is that while the start codon is actually a protein coding codon, it codes for methionine, the 3 versions of the stop codon all have in common that they do not code for anything. They're called nonsense, right? They don't actually mean anything. That's how you just stick something in there that doesn't mean anything, and the ribosome is like, okay, I guess we're done. So Quick question. During translation, ribosomes function to what? Pick the best answer. Great. Fabulous. Yeah. So you can see that they bind the messenger RNA. They bind the tRNA. They catalyze bond formation between adjacent amino acids in order to make the growing protein chain. All right. Now, when mRNA is translated, there's something called a reading frame. You may have heard or may remember this. That's determined by the location of the start codon closest to the 5' end. So let me show you what I mean by that. So here's a generic mRNA. I just made it up. And you could imagine that if you were translating this, right? Remember that that 3 letters of RNA code for 1 amino acid. You could theoretically go, okay, well, the first 3 letters is the first codon, and the next 3 letters is the next one, and so on, right? You could just do it up like that. Or maybe you just start 1 in, blah, 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 blah. Or maybe you start 2 in, blah, 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 blah. But actually how it works is that the ribosome reads from the 5' to the 3' end. So it goes C, A, G, G. And what is it looking for? What is it going to look for as the, now I know where to start? Yeah, it's going to look for the first AUG at the 5' end of the mRNA. So it reads in from this end. It starts here. It says, oh, there's the phosphate group. Let me start reading. Oh, I found my AUG. And then it knows the rest are coding. Okay, so I'm not going to do this with you. This is just something you could do for yourself. Here's the genetic code you might remember from cell biology, and you could be given a sequence of letters and have to translate it. Okay, so we're not going to, there's an exercise in another slide where you would do that on your own. But let me just make sure that we're all on the same page about what we're looking at. So when we look at this table, and we say, here's AUG, here's, other 3-letter codes, AUG or ACA. Are these codons in DNA, codons in mRNA, or are they anticodons in tRNA? What is the table showing us? Great. So this is actually the actual letters that you would find in the mRNA. These are the codons in the mRNA, okay? We know they're not the codons in DNA, right? None of you gave that answer. How did you know that it wasn't DNA? Because there are tables that exist that have DNA and it's not wrong. But how do we know this table is RNA, not DNA? Okay. But when you look at these letters, there we go, Carlo. The U, right? If this was a DNA table, it would be TTT. Okay, it's the U. So we know it's RNA. Now, is it mRNA or is it tRNA? Well, we just have to know that the convention is that they show you the mRNA one. Okay. Now, with that said, there is we can figure out what the anticodon would be. So let's look for tryptophan. Here's tryptophan. This is what it is in the mRNA. If you were reading the mRNA and you were the ribosome and you came across UGG, you would know that was tryptophan. What would the corresponding anticodon be on the tryptophan tRNA? This is 5' UGG 3'. So what would the corresponding anticodon be Good. Remember, the anticodon is the complement, and don't forget the complements are in a reverse orientation. And don't forget that the complement is going to be RNA because it's tRNA, so it shouldn't have any T's in it. There we go. Good. So the tryptophan codon is 5' UGG 3'. The anticodon will be the complement. It'll be a reverse orientation, so it'll be 3' A, C, C, 5'. And let me just show that to you in a little cartoon. Okay, so here we have the mRNA. I just made up another mRNA. Here's the 5' end. There's an A. Here's the first AUG, and the next codon would be UGG for tryptophan. Here's my tryptophan tRNA, little clover leaf. The 3-letter sequence that binds to this, right, bringing tryptophan with it, is the complement to that. Let's suppose that a cell had a mutation in the tryptophan tRNA gene, so that the tryptophan tRNA cannot bind its amino acid tryptophan. What do you think would be the consequence? Take your best guess, and then we'll look at a cartoon to make sure we understand. Good. Good. Excellent. All right, the majority rules happen to be accurate this time. Okay, so let's see why that is. So here's the regular situation. There's our mRNA, there's our tryptophan tRNA plugging in, bringing tryptophan with it. Here's the mutant, okay? So same mRNA, here's my tryptophan tRNA, still able to plug in here, but not bringing the tryptophan with it. As a consequence, this becomes a stop codon. It communicates to the ribosome the same way as if this was nonsense. What happens in the case of a stop codon is there's no tRNA to plug in here, so that means there's no amino acid. And so the one that was just over here has nothing to bind to and nothing to connect to. And eventually ribosome's like, okay, well, I'm not going to wait around forever. And it terminates. When this shows up with no tryptophan attached, the same thing happens. And so the ribosome just stops. It doesn't skip over, right? It still could complete proteins that have no tryptophan in them. It certainly can still initiate translation because the methionine tRNA is fine. But it wouldn't be able to complete proteins. They would be shortened if they had a tryptophan in them. All right, here's the exercise that I'll leave with you over the break just for fun so that you can test yourself. And in the meantime, let's take our 10 minutes. You've earned it. I will see you at 11:50 to keep talking about genetics. Okay, we'll see you at 11:50, and I'll leave this up for anyone who wants to do it. All right, good friends, let's talk about the next important concept that we want to nail down, which is sort of emphasizing and adding to our understanding of the difference between eukaryotes and prokaryotes at the genetic level. So bacteria are different from eukaryotes in that their DNA is a small circle and there's no nuclear envelope. And there are consequences in terms of replication, right? Because whereas in eukaryotes, you have this complicated mitotic process with a spindle and the nuclear envelope dissolves and comes back. And you have to complete one round of replication. Then you've got to make a new nuclear envelope, divide your cell, and only then can you go on to the next round. That's not true in bacteria. They do something called continuous replication. They start the next round of replication when the last round is about halfway finished. So daughter cells in log phase inherit 1.5 chromosomes. So they can get the rest of that chromosome completed in half the time. Now, there is another issue that comes up, or another difference that's important between eukaryotes and prokaryotes, that is a consequence of no nuclear envelope. When we think about a eukaryotic cell, transcription happens where? Where does transcription happen in the cell? Exactly right. It happens inside the nucleus. And it must be fully completed. That mRNA then has to leave through the nuclear pore and enter the cytoplasm where translation takes place. So they happen in different compartments, and there has to be a full, like you have to complete transcription before you can do translation, just like you have to complete replication before you can start again. Is that true in bacteria? Well, where does transcription occur in bacteria? That's right. It happens in the cytoplasm, which is the same place as translation. So the consequence of that is something called simultaneous transcription translation. There is no need to wait for the transcript to be finished before you start to translate it. So let's see this in a picture. Okay, so here is a eukaryotic cell, and there's the nucleus, and we're going to put a piece of DNA in it, and then we've got our promoter. There's our gene. RNA polymerase reads it and makes a transcript. RNA goes out into the cytoplasm and is translated into protein. Okay, so we do this, and it has to leave, and then we get the ribosomes doing their job. But is that what happens in a prokaryotic cell? Well, here's the DNA, there's the promoter, there's the gene. Okay, RNA polymerase gets started. It's made a little transcript. The first part of it is the 5' end as it's coming off of the RNA polymerase. The RNA polymerase is not even finished yet. But this 5' end can be grabbed right away by a ribosome because they're in the same compartment. And it can start translating before transcription is finished. Okay, and by the time we are finished transcription, we've got multiple ribosomes to go and work into town on this one, and the same thing has happened on other transcripts. So we can see that here, okay? This thin line that you can barely see that in the cartoon is purple is the DNA in the cytoplasm of a bacterium. Then these little blue things are the mRNAs, and then all these little jelly beans are or beads are the ribosomes. So we have translation happening while transcription is happening. They're not consecutive, they're simultaneous. And can you have simultaneous transcription translation in eukaryotes? No. I mean, not as far as the cellular DNA is concerned. So what do you think is like the ultimate, like big picture consequence of this? Think about a eukaryote and a prokaryote that both have the ability to, let's say they're both facultative anaerobes, okay? So they can live in oxygen and then they can switch to something else if oxygen goes away. And let's say they even do the same type of fermentation. Let's say they switch to the same process. So they're happily in some oxygenated environment together, and then boom, the oxygen goes away. Who's going to be able to switch to anaerobic metabolism faster? That's right. All things being equal, it's going to be the prokaryote who's more agile, who's able to pivot more quickly in response to changing environmental conditions. Because once the cell realizes, oh shit, I've got to do something different, and I don't have the enzymes to do that thing yet, but I do have the genes for it, and it goes and says, hey, go make some protein for me real quick, that can happen faster because we don't have to wait for this transcript to be made before we can start to translate it. Okay. All right. So now we come to the fun part, which is gene expression, which we just talked about, transcription, translation, is regulated. So in every cell, this is true. In human cells, in a way, it's easier for us to sort of comprehend this because, you know, you've learned a little bit, like every cell in your body has the same DNA, right? except for red blood cells. They're a weird exception, but never mind. But you know, your original DNA is, you got some from mom, you got some from dad in the zygote, and then that cell was copied and copied and copied, and all the cells in your body have the same DNA. But they're all different tissues, right? The heart cell and the muscle cell don't express the same proteins. So that genome that they share in common is not used in the same way in every cell type in your body. Gene expression is regulated. The cell kind of pays attention to which instructions am I carrying that are relevant for me and ignores the rest. Now, when it comes to bacteria or single-celled microbes, they're not part of a multicellular system. They don't have, they're not sort of regulating their expression to do specific things because they're a particular cell type, but they are regulating their expression to respond appropriately to their environment. Right? Oh, the temperature went up a little bit. I've got to do something different here. Or the glucose ran out, but there's still lactose. I got to do something different, or whatever that change might be. Okay. So expression of genes in all cells is regulated. We don't express every single gene that we possibly could into protein all the time. And the reason for that is, you know, there's genes in here that are going to be useful in some contexts and not others. And it would be wasteful to make everything. It costs energy to make protein, and energy is limiting. And the more you waste it, the less kids you can have, and the less successful you'll be evolutionarily. So you only make the proteins you actually need, which means you've got to have some kind of system to say, let's put the brakes on this one because we don't need it right now, or let's ramp up the production of this one because we do. All right, so we need some definitions to talk about genes. The genes in the genome come in sort of a variety of flavors. Some of them are so-called constitutive genes. What does that mean? What does it mean, constitutive, this word constitutive? From the prep, if you remember, what does it mean? You could raise your hand or write it into the chat, either one. I've got my chat open, I got my participants open. Just help me out, guys. What does it mean? Good. All right, so some answers in the chat from Carlo and Libri. These are genes that are always made. That means that they're not regulated, actually. They're just the kind of things that the cell is pretty much always going to be in need of as long as it's alive. So there's no point in turning them on or turning them off. They're constitutive. They're transcribed all the time. Can you think of any examples of things that you might predict would be transcribed all the time? Things that the cell might need, like in pretty much every environment. Yeah, Leslie, tRNAs, ribosomes, indeed. Ribosomal proteins, ribosomal RNA, and tRNA are all important for translation. And whatever you're doing as a cell, if you are alive, you are making protein. Once that stops being possible, you will die. So you need that machinery all the time, okay? Now, with that said, there are many enzymes that are regulated because they're only going to be useful sometimes. For example, enzymes that are important for breaking down some sugar and using it for fuel. Well, there's no point in making those enzymes if that particular sugar is not available. Or enzymes for making something, like an amino acid that you need. Well, there's no point in making the enzymes to make that amino acid if you just happen to have stumbled upon a whole bunch of amino acid in your environment. So there are ways to turn on or off the enzymes and structures that the cell need depending on its environment. So genes can either be repressible or they can be inducible. The repressible ones, what is their default state? Are they on by default and then we turn them off? Are they off by default and then we turn them on? What is their default state? Repressible, a word that means could be repressed, but implies that it isn't by default repressed, means yes, these are normally expressed. The default state is transcription is happening. And there has to be something specific chemical in the environment that actively turns them off. These would be the kinds of things that you need by default. And there's only would turn them off if the environment says, oh, whoa, we have enough. Okay. So for example, can any of you think of examples? You know, it's fine to use the simplest one from the prep. Yeah. This is very typically how we regulate anabolic enzymes, building stuff, okay? Especially important stuff, right? The poster child for this that we will study is the tryptophan operon. These are the enzymes that build the amino acid tryptophan. Just like you need all of the pieces of the translation machinery all the time, as long as you're alive, you also need all the amino acids. And so by default, you need to be making tryptophan as one of the 20 amino acids that you're going to use. What would be a good environmental signal for something like that? If we wanted to, why would we stop making it? If normally we definitely need tryptophan all the time. Okay, just like we need all these things all the time. Why would we turn off the enzymes that synthesize tryptophan? What in the environment might force us to do that or suggest that we should do that? If you just ate some, exactly. If you just run into a stash of it, why bother making more, right? Put your energy elsewhere. So anabolic enzymes, such as those that regulate amino acid synthesis, are typically regulated this way. The default state is that they're made, so they're making the amino acids, and then they could be turned off by an environment in which that particular amino acid was high. Inducible is the opposite. What's the default state for inducible? Could be induced. By default, are we getting transcription on or off? Yeah, that's right. The default state for an inducible gene is that it's normally not being expressed. It's something that you actually only need in very particular context that the environment can tell you about. The environment's only going to turn this on if you need it. Can you think of an example? Again, thinking about the prep that you've done, what you've learned about, can you think of an example? Something that would be inducible. Yeah. This is typically how catabolic enzymes are regulated, things that break stuff down for energy. Because catabolic enzymes are specific to a particular thing, like if it's sugar metabolism, for example, they may be the enzymes that break down lactose, or they might be a different set of enzymes that break down trehalose, or a different set of enzymes that breaks down xylose. You don't want to make those enzymes unless what's true in the environment. Unless the thing that you're breaking down is actually there, okay? If there isn't any lactose, you definitely don't want to be making lactose metabolism genes because it's a waste of energy, right? Or lactose catabolism enzymes, I should say. Okay, so again, lactose metabolism genes, the ones that break down lactose, for example. Here's the permease, here's the enzyme called beta-galactosidase. It brings lactose in and chops it into pieces. You only want these expressed if lactose is actually present in the environment. Okay, so now we have to talk about something called operons. You may or may not have heard of these before, but it's typical in prokaryotes that DNA is organized into operons. And it's not unprecedented to see that in eukaryotic cells, but it's not common either. Very commonly in eukaryotes, there's a promoter and a gene. Every gene gets its own promoter usually. And so there are some examples in some organisms of operons, but it's not that common. Prokaryotes is extremely common. And it makes sense, especially when you think about prokaryotes as sort of the theme of the day as being really streamlined and trying to be super efficient. Because if you're a prokaryote and you are, regulating your genes like, okay, if lactose comes along, I want to quickly turn on the enzymes that help me metabolize lactose. Well, it's a little bit wasteful and takes up space to have a promoter and a gene for every single part of the pathway. It makes more sense if, in order to metabolize lactose, you need, let's say, 3 enzymes. Just put them all together next to each other in the same transcript with one promoter that's regulated the exact same way. So when this is needed, we already know these 2 are needed, so might as well make them all at the same time. So what is an operon? An operon is when several genes are co-regulated by the same promoter. And the typical structure of the operon is the genes are right next to each other, right? They're going to be part of the same transcript, and then the ribosome will figure out that they're all different proteins. So there's one promoter. And who sits down on the promoter? Like this is a landing pad for what enzyme? That's right. There's a landing pad for RNA polymerase. So this is where RNA polymerase binds, because it's like, oh, there's the start of a gene. But for regulated operons, you'll notice there's this little space in between, and this is a landing pad for another protein. What's this space in between the promoter and the protein coding genes called in an operon? That gives it its name, basically. Yes, this little space, and it's a series of letters, just like the promoter, and it's a landing pad for a particular protein, just like the promoter is, but it is called the operator. And what is the protein called that binds here? Indeed. It's a protein called a repressor. And I think this part is maybe sometimes hard to integrate when you're first learning this. But unlike RNA polymerase is like a dude, like it's one guy. And yes, there's like 1,000 RNA polymerases all running around, but they're all the exact same as each other. There's 1,000 of them because we need to be in 1,000 places at once, but they're basically all derived from the same gene, right? The repressor is specific to this operon. So if this is the operator for operon, let's say these are genes that code for proteins that metabolize lactose. This is the operator that binds the lactose repressor, and it doesn't bind other repressors. Okay, now to sort of like bring that home, usually the gene that expresses this protein, that's the instructions for this protein, is somewhere nearby, like close by, actually in the genome, but it has its own promoter, and that promoter is constitutive. You notice that there's no space between the promoter and the gene. It's not regulated, it's constitutive. Meaning what about this repressor? It's constitutive meaning what? Yes, Mackenzie. Yes, Spencer. Yes, Sam. Yes, Libri. That's right, Carlo. Yes, everybody. It's always being made. This repressor is always around. The job of the repressor is to bind the operator. And when it does that, you'll notice it's in the way of the RNA polymerase. RNA polymerase would normally move forward and transcribe all of these genes so that we can get to the business of making proteins. But if the repressor is in the way, it behaves like a boulder or a cow on the train tracks, right? Prevents RNA polymerase from moving forward. All right, so we've already done this part. Let's move forward to how repressors work. So here is an operon. It's got 3 genes. There's this region here that binds the repressor. What's this region called again? The repressor binds where? What is this? That's right. That's the operator, and the repressor binds the operator. Then we have this region over here that RNA polymerase binds to. What's this called? Correct. All right, so RNA polymerase binds the promoter. The repressor binds the operator. And if this is what's happening, okay, then are we getting transcription, yes or no? Right, if the repressor is bound to the operator, if the repressor is doing its job, if the repressor is active, then RNA polymerase can't transcribe and we don't get transcription. So repressors, when they're active, shut down transcription. So this is one of the beginning of a series of double negative logic circuits that you kind of have to draw a picture if you're going to understand it, because most people, now I'm not going to say I know you, but most people have a hard time keeping this logic circuit verbal in their heads. So a picture is worth 8,000 words in this context. Okay, so here what we can see is there's the gene for the repressor. We can see that it's constitutively being made, and it's always being made into a repressor, and the repressor is sitting in the operator and transcription is not happening. If this was the end of the story, it'd be pretty boring, right? Because if this is the end of the story, well, no, there's no transcription ever. So why would we even bother even having these genes? But that's clearly not possible. This is a allosteric DNA binding protein. DNA binding, here it is, binding DNA. Allosteric meaning, anyone know what this means? It's also a word we used when we talked about competitive and non-competitive inhibition. It means that it has the possibility of another shape. The allosteric proteins exist in two alternative shapes, and the way to change their shape is to bind to them and cause that shape to change. So something else has to bind, okay? And when that specific special something is able to bind at an allosteric site, it causes the shape to change. Now, remember this happened for enzymes, right? Enzymes have an active site where the substrate binds, but if they're allosteric, they have a second site where the allosteric regulator can bind and change their shape. So when this gets changed, when its shape is changed, it can no longer bind the operator. So that's how we get it to go away so we can finally get transcription. So the function of the repressor in this active shape, what I call an active conformation, an active shape, the function of the repressor in the active conformation is to bind to the operator and prevent transcription. Okay, now, before we move forward, I just want to say something up front that I hope sort of lays the groundwork, which is that whether this is an inducible or repressible operon, the most important distinction between those two types is what the repressor is like when it's born. Okay, all repressors have this allosteric nature. They have one shape that does bind the operator, they have another shape that doesn't. And in order to change them from one state to the other, something can bind them. Some environmental chemical cue can cause that change. But some repressors, as shown here, when they are constitutively made, immediately assume the correct active conformation. That is to say, right out of the gate, they are born ready to bind to the operator. And some of them actually are born in the inactive conformation. They're born lazy, okay? And in order to get them to sit in the operator, you actually have to have the environmental cue work on them. So some repressors are born ready, needing no further information. By default, they go and sit right here in the operator. If you want to get them off, an environmental cue binds them and removes them. And some are born lazy in the inactive state, and those cannot go here unless an environmental cue binds them and changes them to the active shape. So does that distinction sort of clear at the beginning? Okay. So we're going to lean pretty hard on that. And you may find yourself going, wait, am I born ready? Or am I born lazy? I don't know. I don't remember. So let's see if we can get there. All right, so let's talk first about the ones that are born lazy, okay? Some repressors are not born ready. In this picture, this repressor is born ready. If you want to get him out, you got to get some kind of environmental cue, has to come and bind him and change his shape. And then he's like, I like to think of it as like a, you know, a friend or like a donut or a coffee break comes along and says, hey, dude, you've been working really hard. Go take a break. Here's a coffee. And he goes off and puts his feet up for a minute. Okay. This is that, you know, everyone knows these types of people, right? This like type A person who never takes a rest unless you bludgeon them over the head. And then others of us are quite content to sit on the couch eating potato chips and watching TV or playing video games until someone kind of takes a shovel and shoves us off the couch. So repressors that are born lazy, they're born in this shape, okay? They're constitutively made, so they're always being made, but they're born in this wrong shape to bind. They can't do their job. They're like, it's very cozy in my mom's house. She does the laundry and makes me sandwiches. And then if you want to get them into the operator, if you want to make them do their job, you've got to actually give them something, give them some kind of incentive, or you've got to give them some kind of motivation. Okay, so born lazy needs some kind of incentive to actually prevent transcription. Now, with that said, I want you to think about which scenario you would want for the different types of genes. Okay, so let's go back here. If you're born ready, what's the default state for transcription? Off or on? Okay. If you're born ready, that means you're binding, which means there's no transcription. Transcription is off. Remember I said it was an inverse relationship. Annoying, isn't it? When this is doing its job, this can't. Okay. If the repressor is active, transcription is not. Does it make sense to do this kind of repressor for an inducible or a repressible gene? Yeah, inducible, right? You want by default this to be off, and the inducer, the environmental signal, is what's going to take this guy and tell him to go for a coffee break so you can sneak in some transcription. Let's look at this one. Here we have the guy that's born lazy. Okay, so what's the default state if this guy is born lazy? Transcription on or transcription off? Right. If he's born lazy, he's not going to be sitting here in the operator, and RNA polymerase can transcribe all day long. A big party at RNA polymerase's house. Okay. So there has to be an environmental co-repressor that convinces this guy to actually sit in the operator and turn off transcription. So is this how you would regulate an inducible or repressible operon? Exactly. Yeah, this is how you regulate a repressible operon. Because repressible, remember, we want the default state to be transcriptions happening. Only when there's some kind of environmental cue do we want to shut things down. Okay. So just a reminder, a repressible operon is when transcription is usually on, right? Transcription is usually on. Something in the environment can turn it off, okay? Such as anabolic synthetic enzymes that build stuff like a really important amino acid like tryptophan. An inducible operon is usually off. Something in the environment can turn it on. This is how we regulate enzymes that metabolize what we would call non-optimal carbon sources like lactose. Now, here is just going to require a little bit of memorization, which is to say that the best carbon source, the favorite carbon source, most microbes, and that's true of your body too, is, of course, What is it? What's our favorite carbon source? Okay, which one, Jolene? Yeah, that's right, Carlo. Specifically glucose. Think about respiration and how the entire thing starts the funnel where glucose is at the top. So you're kind of like primed for that. So most microbes are like you in the sense that their favorite carbon source is glucose. And this is, again, this is kind of oversimplifying the situation, but nonetheless, it's true in a lot of cells. And other things can be used if you have the right enzymes, but aren't as good because you don't get as much energy because you've got to modify them somehow, which costs energy in order to put them into the glucose track. All right, so let's see if we understood the repressible inducible logic. Here we have a situation where there is a repressor that's born ready. It's born, it's constitutively made, boom, it sits in the operator. Transcription is blocked. If you want to get it to move and get transcription going, there has to be some kind of environmental signal that binds the repressor, changing its shape so it can't bind the operator anymore. Is this how we regulate repressible or inducible operons? Correct. All right, inducible. All right, so here's another situation. Is this repressor born ready? Nope, he's lazy. He's sitting on the couch, munching on Cheetos, which sounds really delicious right now. I'm on a 75-day challenge with my wife to not eat junk food, and I'm telling you, potato chips sounds like I could maybe sell my grandma for that if she was still alive. Okay, so repressor, not born ready, born lazy, not binding the operator. So that means we do get transcription. By default, transcription's happening. If we want to stop it, there has to be some kind of environmental co-repressor to bind the repressor, change its shape to the active conformation, get it to sit down, do its job, and stop transcription. So is this how we set up repressible or inducible operons? Great. That is correct. Repressible. Okay, so we're going to make a little headway into this material. And we're going to start with what's actually the simplest thing actually to talk about, which is the tryptophan operon. The operon includes the genes to make the enzymes to synthesize tryptophan. The genes to make the enzymes to synthesize tryptophan, an amino acid that you always need in order to be ready for whatever the world can throw at you. Now, you may actually know this from nutrition, or maybe A&P covered it, that tryptophan is one of the amino acids that we actually can't make. We have to have it supplied in our diet. It's an essential amino acid. It's one of our growth factors. But many microbes, including E. coli, can make their own tryptophan using glucose as a starting material, as long as they have some nitrogen handy. However, this process of tryptophan production is costly. It costs energy. So if tryptophan was present in the environment, should E. coli transcribe these enzymes or not? Right. If tryptophan is actually plentiful, we don't want to make more of it. But absent that environmental cue, absent an active presence of tryptophan, what should the default state be? Yeah, we want our default to be, let's make tryptophan unless the environment says, whoa, 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 you've already got enough. So the default should be, yeah, let's make these enzymes because we need tryptophan. But if the environment contains tryptophan, tryptophan can be this co-repressor, this cue that prevents transcription from happening. So is this a description of a repressible or inducible operon, do you think? Right. This is repressible. It's by default on, but can be turned off by this environmental cue. All right. Now, if what we want is a repressible operon, then what kind of repressor do we want? Do we want one that's born ready or lazy? Do you want it to be born active or inactive? Right. We want the default state for that repressor to be inactive, so it's not doing its job, so it's not sitting in the operator, so it's not interfering with transcription, which should be born lazy or inactive. Okay, so let's have a look at that. Here are the 5 genes in the tryptophan operon that encode the enzymes necessary to make tryptophan. Here's the operator, the repressor combined there, the tryptophan repressor, I should say, promoter, where RNA polymerase binds. Here's the gene to make that repressor. We can see it's constitutively made, but in the inactive conformation. So transcription's normally happening. And the reason transcription by default can be on is because this repressor is born lazy. What change in the environment do we want to be the signal to shut this down? What chemical or environmental cue do we want to shut this down? We're making these enzymes to make tryptophan. We're making tryptophan, making tryptophan. Yeah. If we run into a stockpile of tryptophan, or conversely, we've been making a bunch of tryptophan, but the proteins we've been making haven't needed it, and so there's an excess. We have extra. For a second, we want to stop making it so we can use up, you can kind of eat down into those reserves so we're not wasting energy making something we don't actually need. Okay, and so here's our born lazy guy. We want to convince him to go sit on the operator. We got to give him something. What is it that makes the most sense to be the chemical that directly binds to this repressor? We want to shut this down when tryptophan is high. So the thing that's the simplest that works really well is for tryptophan itself to be the inhibitor, or the co-repressor, I should say, the activator for the repressor that causes it to actively repress transcription. This is why pictures are helpful, guys. So that's what tryptophan does. Now, and the elegant part about this is this relationship between this chemical and the repressor isn't like glue, okay? Like many relationships between chemicals and proteins, there is a little bit of like, for a second, this guy might go take a potty break or something, and he wanders off for a minute, and then this guy starts to get lazy again. But there's so much tryptophan around that while he's off on a potty break, some other tryptophan is like, boom, I'm gonna sit right back on you, get back to work, dude, you can't go yet. And as soon as we start using up the tryptophan, like when we've needed it, and it starts getting less and less in the environment, when this guy goes to take a potty break, if there's nobody to replace him, there's enough time for this to revert to this, and then we get transcription again. Okay, so let's see if we understood that. When tryptophan is not present at high levels in the environment, what state is the repressor in? Active or inactive? Right. The repressor is inactive, lazy. And so as a consequence of that, since he's not sitting in the operator, what's going on with transcription? That's right. Transcription's happening. Okay. Now, let's add some excess tryptophan, right? We didn't need as much as we thought, or we ran into a stash of turkey legs. Got a lot of tryptophan. What does it do to the repressor? Yeah, it activates it. Now it goes and sits in the operator. And what happens to transcription? Boom, shuts off. Good. That's right. So you can see that here, okay? Here's the default, inactive, not doing anything, playing, I don't know, I'm not a video game person, so insert your favorite video game here. And then, so we get transcription, which means we're getting tryptophan. And then boom, we've got extra tryptophan because we haven't used it up or because we found more. Turns on the repressor, goes and sits in the operator, stops transcription long enough for this stockpile of tryptophan to go down. All right, so let me just kind of look at our time here. We do have time, so I'm going to blow past this and talk a little bit more before I turn my attention to the N180 project. All right, I want to do a little comparison to the lactose operon. So unlike the tryptophan synthesis anabolic enzymes, the lactose operon genes are catabolic. They are what's required to break down the sugar lactose. And so you have the 3 genes, you've got the operator. Who's going to sit here in the operator? A repressor will. Is it going to be the tryptophan repressor? Nope. What is it going to be? What would we call it? This is the lactose operon, so we're going to call this the lactose repressor, or the lac repressor. Good. So there's a protein that's going to sit down here, but it's its own thing, right? It's made by this gene, actually. There's the promoter or RNA polymerase is going to bind. When do we want these to be made? Do we want these to be made by default? No, we don't. We only want them to be made in the case where there's an environmental cue that says they should be made, right? When lactose, for example, is actually present. So how do we want to set this up? Do we want this to be inducible or repressible? Right. We want it to be inducible. We want the default to be you are not being made unless the environmental cue of lactose, for example, tells us to turn you on. Okay. If that's how we want to set it up, if we want an inducible operon where normally there's no transcription, then how does our repressor need to be born? Should it be born ready or lazy? Should it be active or inactive? Yes. We want our repressor to be made in the active form. And that's how it works, okay? This is normally how the lactose operon is. It's normally repressed. Here we've got our RNA polymerase on the promoter, but over here we've got our repressor because when it's born and it's constitutively made, so there's lots of this all the time, and it immediately goes and sits on the operator. This is like, you know, pops out the womb and puts on a suit and just goes straight to work with a briefcase, freaking out its parents. Okay. How do we get this repressor to go away? We need an environmental cue. What should that environmental cue be? What should the chemical be that binds this and removes it? That's right. We want to get rid of this guy if we want transcription to happen, but we only want it to be happening when we actually need these genes, which is going to be when lactose is present. And so you can see that here, okay? So lactose is the inducer. Here's our guy born ready. If lactose is present, it binds this type A security guard or whatever, says, dude, take a break, relax before you have a heart attack. You know, hands him a cup of coffee and a donut, says, go sit in the corner for a minute. And because he's not at his post, RNA polymerase can do his job. All right, so when lactose is not around, what is the repressor doing? Is it active or inactive? Right. Yeah, think about it. Some of you were like, wait, the first thing I said was wrong. So this is inducible. We only want these genes to be made when lactose is present. So we want our repressor to be born ready so that transcription can be off. When lactose is present, what happens to the repressor? The association that I have, which I will give to you, but I warn you, it's not for everybody. Lactose is in milk, which is like cream in coffee. So you hand your coffee to the security guard to make him sit down and relax. Okay, so when lactose is present, the repressor does come off of the operator. That happens. That's true. Okay, what I'm about to tell you next does not invalidate that. But what I'm about to tell you next is that transcription does not immediately ensue in this circumstance. And that's because in these inducible genes, there is often an additional level of regulation that's required before transcription will happen. So the first, like this part and this part all kind of like is sensible, but this part requires more explanation. If you've done the prep, you know what that extra thing is. What is that extra thing? Either environmentally or you can talk about it mechanistically. You can say what in the environment also needs to be true for transcription to resume, or you can say what other, transcriptional regulator protein needs to be involved, if you remember from the prep. Just put it into the chat. And if you have no idea, put a question mark. That's a great contribution. Yes, that's right, Carlo. In addition to lactose being around, which does remove the repressor. Just notice this is a typo, so sorry for those of you who are kind of anal like me. If this is bothering you, I apologize. In addition to removing the repressor, which lactose will do, there needs to be an activator called CAP that comes to the operon as well, and that only happens if glucose is gone. Okay, and so this is really where we're going to break. Hopefully I've like intrigued you enough if you didn't already know what I'm about to tell you. Hopefully I've intrigued you enough that you're going to go back and look at that prep before our next class so that the class we do on Monday will not take forever and won't be as painful, because we do need to finish genetics and then go on to viruses. So let me go back to what I wanted to do. I'm going to move some things around here. And pop this in here. All right, so do go back and look through that stuff again, or look at it for the first time if you haven't had time, so that you're mentally prepared for what I'm gonna put you through on Monday. I want to take a second to talk about the Unknown AD project again. But before I do that, I want to do your to-do list, because some of you have heard this already and I don't need to go through it twice for everybody. So if you don't need the extra guidance and you're ready to go, I can let you go. So let's just briefly talk about what's coming up urgently in the future. So tomorrow is the Unknown ID checkpoint 2. Many of you have done this already, so good job. And I've graded what I saw yesterday. There may be some new submissions today. So get this done, submitted. Now, with that said, to somewhat relieve the pressure, I am accepting this until Sunday, but you really want to get it done as soon as you can, because summer is so short. And step 3 is really where it's at. Sunday, we've got a metabolism review quiz. You've got the first part of lab 4, the digital part. You've got the post-lab quiz, because once you've done the digital part and done the background and all that stuff, really, you've got all the information you need to do post-lab 4. So let's get it out the way. And pre-lab 5 to prepare for transformation on Tuesday. To prepare for Monday, make sure that you do have a chance to review or for the first time view the lactose operone stuff and do the bacterial genetics worksheet. Also, definitely do the virus prep. Take that time this weekend, so important. Viruses are super cool and fun, but there is another complicated genetics component when we come to life cycles, and it's super sucky to have not done the prep for that part. Okay, so before I go to my official Q&A, well, maybe I will do the Q&A officially, just so that if there's people who want to ask questions right now, you can, and then if you don't have any, you don't want to talk about the unknown ID project part 3, you can take off. So what I'm going to do now is take some questions for a few minutes, and then I'm going to briefly go over unknown ID 3. And if you're done right now and don't have questions, then log out. Goodbye. If you do have questions, raise your hand. I'll get to them. And then after that, I'll just go over unknown ID part 3 with anyone who really needs to right now. Aubrey says, have you adjusted lab 4 since we weren't in lab this week? I have. So you mean like I've added the digital component to the lab 4A, and I haven't adjusted the deadlines because lab 4A is still due, but you will have an extra week to do lab 4B. But the post lab is still the same deadline. Does that help? Does that answer your question basically?
Yes, it does. So we're only submitting lab 4A this Sunday?
Yeah. Then there's 2 components to it. So there's the case study that was already posted, and then yesterday night, very late, I added the catalase oxidase nitrate part.
Okay, sounds good. Thank you so much.
You're welcome. Okay, any other questions, guys? Carlo had that same question. Carlo, is your question answered, or do you have any follow-up on that? Okay. Riley, you had a question. Go ahead.
It might just be as simple as this, but I followed my dichotomous key down, like asked for the catalase test, got the results back, and then followed the endospore stain and got narrowed down to just bacillus. That's just the bacterial group. I stopped there. I'm done with part 2. Turn that in, move to part 3.
Yes.
Okay, that's all I was like, I feel like it's too easy, but it might be wrong. I don't
Don't
know.
worry, Riley. Step 3 is going to kick your butt, so don't feel bad. You've done what you need to do for step 2. Just, yeah, buckle up for the rest of the ride.
Thank you.
Any other questions, guys? All right. Thank you, Spencer. Not yet, but I'll get there. All right. So if you want to know about unknown ID part 3 or have questions, then stick around. If you don't, then just sign out and have a good weekend. Make sure to do all the work that I just left you with. But I'm just going to go back to my slides and see who wants to go through this part. Okay. So just so I know, if you do want me to go through the unknown ID step 3 instructions, just raise your hand so I know that there's anybody who wants to do that, because we don't have to. Okay, great. Riley, it's a good time for you to do this too. And Jolene, yes. Okay, perfect. Let's do it. Okay, so and I'm recording this so that everyone, hopefully, if you're reviewing, you'll see this. But you want to make sure you read the instructions and watch the video. It's super important. You'll have questions after that, most likely, but do read the instructions and watch the video. If your group is Enterobacteriaceae, pay attention to my initial recommendations. There's 4 tests that I suggest you start with. All the other groups, however, it's really up to you. Big picture, all of you are going to be doing a process of elimination where you request metabolic tests, eliminate species in your group that have the opposite result of the test result that you get, leaving you with a smaller pool of candidates that gives you a way to figure out what your next test should be. And you continually serially eliminate until you end up with just 1 species. It's also possible for you actually to end up with these 3 species that are not distinguishable by metabolic testing, in which case you should email me because there is something you can do, but it's going to take you a minute before you get to that point. So how do you choose your tests and what are you choosing them from? First of all, you're going to the Bergies. So let me see. I think I want to share my screen here. Whoopsie doodle. I want to share my screen, not with the, oh man. There we go. I want to share my screen so that you can see where in the course shell to go. So once you know your group, you just scroll on down to the Burgess manual. Oopsie doodle. And the Burgess manual is a multi-volume series of hard copy books that cover all bacteria known to man. I've excerpted PDFs from them. So your group will be one of these. Let's suppose, let's go with Bacillus because Riley was Bacillus. Let's do that one. So when you look at this, you'll see it's like an 8-page document that has all your species across the top. So there's there's like 15, 30, 7. Well, they have numbers with them too. And all these pages have the same things down the side, but they have different species up top. So you have about 100 and some species, and you've got all of these different tests. And it's showing you for this species, a catalase test would be negative. This one's positive. For this line item says, can they do fermentation of arabinose? Yes, yes, yes. No, no, we don't know. Perhaps, not sure. So it gives you the results that are known for all these species. If you're not Enterobacteriaceae, then you have no specific instructions about where to start. Okay. So you have general instructions for where to start. So let's go back to the general instructions. This is true for everyone. Eventually, even if you're Enterobacteriaceae, you're going to eventually end up in this position. So the first thing is, unlike part 2, you can order more than 1 test at a time. You can order up to 4 tests at a time, no more than that. And you want to be careful not to over order. So the number of tests you can order at one time depends on how many species are in your group at that moment in time. Entero and well, pretty much everybody starts with enough species to order 4 tests as their starting point. But once you order those tests, get the results and use them to eliminate candidates, you will have fewer candidates in your pool. And if that number has dropped to 30, then only order at max 30, but only order at max 3 tests at a time. You don't have to. You can order just 1 at a time and be safe, but don't order more than that. If it drops to 20 candidates, 2 tests, and if it's 10 or below, then 1 test at a time. So how do we pick these tests? Again, if you're Enterobacteriaceae, I tell you what the first 4 tests would be. I don't tell anybody else just because there's 4 obvious ones in Entero and there's lots of good choices for the other ones. But regardless, at some point, you're going to have to make that decision on your own. So let's look at how and what would make a good test versus a bad one. You want to pick tests where, when you look through your candidate pool, there is a decent proportion of pluses and minuses. So, you know, when you look through even just this first page, almost every test has some pluses and minuses. So almost any test in the beginning is going to be fine. But there's going to be some that are maybe better than others. Like, you know, it may not be the case that when you look through all 8 pages of this document, like there might be lots of minuses later in the document, but right now, this 2% salt test, there's only a couple guys that are negative. Most of them are positive. This would be a fine test to order, but is it the best one? I might personally go for one that was more 50-50 minuses and pluses, you know, but it's kind of your call. Also be careful not to order tests you've already ordered. For example, for Bacillus, in order to get into the group, you did a catalase test. You did this test already, so you don't need to reorder it. You can just use it. This group, catalase is defining for this group, but there are some species that are exceptions, so you can immediately roll those out. You can be like, well, I was catalase positive, so I can immediately get rid of these guys that are not catalase positive. Riley.
So you said that we have to be like, I guess, good at eliminating and like show how we did. Would it be acceptable to like say print this document out and then like as the tests go on, like say I already ordered a catalase test, so I can go through and highlight like this was positive, like whichever ones were positive, or would that not be a good way? And then like at the end, like attach that to my journal, just like showing kind of my process of elimination on that sheet, or is it more like handwritten?
Yeah. So I will answer, there's sort of two answers to your question. How you annotate the process of elimination, how you exactly annotate it is up to you. I will show you my favorite way, okay? And this is common, most people do this, but it's not the only way, and you're not married to this. I will show you how I would actually do the elimination so that I could remember what I did and, you know, what I found works the best. In terms of what needs to be in your lab notebook, separate from this documentation process of elimination that makes sense to you, however you want to do the circling and crossing off, you have to write it out in words in your lab notebook. So I'll cover both those things. Those are really good questions. Let me start by going to doing like a quick show and tell for how I would do this for myself. Me personally, not the notebook necessarily, but just like, how do I keep track of everything the simplest? And again, your process might be very, let me share my screen in a second. Share. Okay. I'm just going to make this a little bigger. I'm gonna write on this. So pretend that you had a hard copy of this. And I'm just going to use my pen to write on it. Okay. And let's start with the catalase test. You know, I know it's positive. You could even put the date that you got it if you want. That's not necessary. And you go, oh, look, there's a negative, and circle it. That tells you this species conflicted with my result at this test. And then I just go, okay, well, dude, you're gone. And I just cross them out. All the way down. And then I go across and do that. And this would be the same strategy I would use for future tests that I order too, where he didn't agree with me. Now, I'm only doing this for this one page. You would obviously do it for the entire page, right? You do it for all 8 pages. Notice what I'm not eliminating. I'm not eliminating guys that have no information because I don't know enough about them, or guys that have weird letters because I don't know enough about them. I'm not eliminating positives because they agreed with me, okay? And at this point, let's suppose this was my entire candidate pool, which it's not, but let's suppose that I was. Now I've got to pick a test that makes sense, okay? If this is how many I had, I'd only pick one test or maybe 2. In your case, Riley, you'll actually pick more than that if you want to, because you'll have a bigger candidate pool to look at. But if we just use this as a tiny example, what test would you, when you look through this, you go, huh, what test do you think would be a good one to do that has like lots of pluses and minuses? Anyone who's looking at this with me can also, like Jolene or Esther or Riley, can write something into the chat like, what about we do this test? Would that be a good one to do?
I was just looking at the one like 2 below it. I think it's anaerobic growth. It seems like there's an equal amount of pluses and minuses, minus like the couple unknowns.
Yeah. So anaerobic growth, how do we test for that? We're trying to figure out, does it grow without oxygen? What test do we have that would allow us to test, do you grow without oxygen? That we covered in lab 4, we covered in, or no, we covered it in lab 3, and we covered it in the, yes, Jolene said thioglycolate. So you could order a thioglycolate, right? Not only would you get actually anaerobic growth answers, but you get aerobic growth answers too. So you're going to look at that thioglycolate, look at the bottom and go, did it grow there? Yes or no? So let's just, for example, let's suppose you did order a thioglycolate. Okay. So first person in the chat to just postulate what came back. Did it come back as facultative anaerobe or obligate aerobe, or obligate anaerobe? What happened? Facultative. Okay, so here's my tube and it grew all up in there. So what that means is that it's positive for aerobic growth. Notice I'm annotating that on here so I can see it, and positive for anaerobic growth. So it's positive in oxygen, positive in not oxygen. So I can do eliminations for both of these. I can look across here and see if there's any minuses. Oh, there's one. So that guy's gone. Bye-bye. That's all on this page, but you could keep going for the other pages. And then I look for guys that are minus. So that guy's a minus. This one I don't have to circle because he's already gone. So I don't care about him anymore. Minus, minus, minus. So I circle all of those guys. I know I'm really sloppy here, right? So I just eliminate, eliminate, eliminate. You're going to be nicer in your drawings because you're not using a mouse, presumably. Here's another guy who's gone. And another guy who's gone. And you just, so do you get the logic of this? Okay. And then Jolene, did you have a follow-up question?
Yes, I do. So I have Staphylococcus, and on mine, I already ordered the thioglycolate test, so I already have the results for that. And on mine, it has pluses and minuses, but it also has a plus sign with parentheses around it, and it also has, it says like negative W
Yeah.
on it for
So
some of those.
anything that's not a crystal clear minus plus, I wouldn't eliminate on that basis. I would kind of hold off because there's a bunch of annotations in the Bergies, and I don't know all of them off the top of my head. So it's safer to keep them in your candidate pool than rather than getting rid of them if there's any sort of ambiguity.
Sure. Okay, cool. That makes sense.
Esther, what was your question?
So how do you know when you get like to the end of?
How do you know when you get to the end? Good question. Okay, so one obvious cue would be if, and again, I'm doing this really small example, right? I'm pretending that this page is all I have. When if you're in Bacillus, you know you have multiple pages. But if there came a point where there was just one of these that hadn't been crossed off, you definitely know who you've got. Or maybe you have 3 that haven't been crossed off, and when you compare them to each other, there isn't a test where they differ from each other. Okay, that's when you know you're done. Does that help? Well, you're not done yet. If you still have multiple ones at that point, then you reach out to me and say, I have 3, but they can't be distinguished from by testing. So what do I do now? Does that make sense? Okay, great. So you just keep going for, and whatever your remaining candidate pool is, that's how you decide which is the next test to order. Like the guys that are already gone, then they're gone, right? So if you try to go and say, well, what would be the next useful test to ask for? Just make sure that in your remaining candidate pool, which is now in this example, only 1, 2, 3, 4, 5, 6, 7, 8, 9 species left, that the test that you're using actually does have some pluses and minuses, right? Don't ever order a test where everybody who's left is the same or ambiguous, as you know, there's no definitive plus versus a minus. If that makes sense. So let me go back to my slides for a minute. Oh, I am actually already in my slides. How silly. And I want to get at this next question that Riley asked, or answer her question, which involves this answer. When you annotate it the way that I've just described, or whatever your favorite way is, that's for you. And you can submit it. I encourage you to submit it. But the notebook needs to be a little bit more clearly explained. So what needs to be in the notebook? Every time you order a test or a series of tests, line item, date, ordered, la, 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 test X, Y, and Z. When you receive it, line item, date, received, test X, Y, and Z, here's the pictures, include the pictures, put them in. Test X, I see purple, this is positive, meaning la, 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 whatever it means about the organism. Test Y, I see no color change, meaning negative. So that means doesn't have a whatever, and so on. So you have your interpretations, and then you say, test X eliminated 75 things. Test Y eliminated 2 things. Test Z eliminated no things, okay? I highly recommend that you actually list out by name who you eliminated, because you will have to call that out in the lab report. And so if you've done the work up front, it makes it easier, but that's kind of up to you. Okay. Riley says, if a test we don't know about, we Google and learn about it. Yes and no. So the first thing actually to do is this. Let me go back to the previous slide. Why can't I go back? Here we go. If you look through the list of tests for any group, we have many, but not all of them. So how do you know which ones you can actually request? You look in the micro test reference. So let's go to the course shell again. Pardon me. Let's go to the unknown ID project module. And resources page. Look, there's the microtest reference, and when you look at it, you will find the first 3 pages is what are all the tests we actually have? We have all these tests and all those tests and all these tests, but that's it, right? So if you can't find it in here and you really, really want it, you can ask me, I didn't find it in there. Is it there by another name or is it really gone? You can confirm that, but don't do that until you've actually checked. If it's not in here and I say it's not in here, you can't ask for it. As an example of some things that are not found, the first tests here, like pigment of colonies, we don't have those answers. Cell diameter is too ambiguous. You can't figure it out to a high enough level of degree of accuracy with the picture that you're given, so you can't use that. Spore formation you can use, but spore formation is defining for this group, so it's not super helpful. I think there's like one species that's negative, so you can get rid of him. And shape of spores and stuff like that, we don't have that information. And there's some, there's a few sugars we don't have, and we don't have egg yolk reaction or see what else we don't have. We don't have xyloside, for example. All right, let me go back to the slides. I think this is the right place. Yes. Okay, so notebook requirements, I hope are clear. With that said, let's just see if there's questions about this. Did you have follow-up questions, Riley, about if there's a test you don't know about? I have a follow-up answer, but I'll make sure I'm giving you space to follow up first.
No, I think it's pretty clear. I'll probably just go through like the micro testing once first. Would it be a bad idea to just go through the original table and just cross out those tests that aren't even shown on that sheet?
Sure.
And then print out like a second copy in case I can't narrow it down and then go from there?
Yeah, why not? It's not a bad idea.
Okay.
One thing is you will eventually have to talk in the lab report about what the tests are telling you. And the microtest reference does a good job of showing you what positives and negatives look like, but doesn't really go into detail about the interpretation. Like, what is this telling you about the organism? And so for that, you may have to Google it. I'll show you the primary source for that information is in the lab manual right here. So you can look up a lot of the tests here, not all of them, but you can look up a lot of them in here. And you can also ask me, like, do your own digging. But then if you're still feeling a little bit confused, you can say, well, this is what I found about test X, Y, or Z, but I still feel unsure about what it means.
Are we going to need, I know for like a lot of my other college courses, when we do things outside of the lab manual, are we going to need like references in that sense?
Yes.
Like if I find one that I don't necessarily can't find in the lab manual and I find it through like a different scholarly source, that I just like bookmark that and save it for later and be like, this is where I got this.
Yeah, there's going to be a references list for your lab report.
Perfect.
But make sure that it only includes things that you actually need to cite in order to support the things you're actually saying in your own words, not everything that you used sort of peripherally. So if, for example, you read something that was like useful at the time, but then it doesn't need to somehow be clearly called out in the lab report, then you wouldn't include it in that list, if that makes sense. Because everything in the reference list should have a direct in-text citation next to a sentence that says something that needed that reference.
Perfect. Thank you.
All right, I'm going to stop the recording on this